Now that we've covered the details of DNA structure, DNA replication, and the details of the cell cycle, we're ready to pull this all together to understand how it is that cancerous cells behave differently than normal healthy cells. Remember that this cell cycle is something that happens normally in organisms. In order for humans, for example, to make new skin, in order for them to heal bone, in order for them to be able to replace cells, cells will go through this cell cycle. However, we know that in cancer, what we're really talking about is uncontrolled cell division. If we had to define what was occurring with this cell cycle, we recognize that this cell cycle would be occurring in a way that was not under strict regulation. It would be uncontrolled. What does that mean? That means in healthy cells, the cell cycle is highly regulated. It's regulated by different signals that say go, and the cell cycle will go and will make new cells. But it's also regulated by signals that say stop, so that when a cell should no longer divide, or if we should slow the process, the cell cycle will be responsive. In cancer, then, those stop and goes seem not to work the same way, and in fact, cells will continue to divide unregulated. It also can happen at a faster pace, and therefore we end up with one cell and then two cells and then four cells of these cancerous cells that continue to grow, and of course that's going to develop into a tumor. Let's take a look at some of the ways that this cell cycle is controlled, specifically related to cancer. We know that there are genes that are going to influence how quickly our cell cycle proceeds forward. Many of these genes can say go, and that's going to tell our cell cycle to go. That means cells will divide. But we know that genes also play a role in those stop signals as well. So I wanted to talk a little bit about some of the genes that are involved in these stop and go signals in the cell cycle. The first kind of gene we can talk about is what's called an oncogene. An oncogene is actually a gene that causes a cell to become cancerous. How could a gene do that, actually cause a cell to become cancerous? Well, we have to think a little bit about what genes do. We know that genes are essentially the recipe or the code for how to make something. And so if we now have a gene that is going to code for something that is going to increase the rate of the cell cycle, then that's essentially a gene that is going to tell that cell cycle to go. You may have heard of an example of an oncogene. When we look at breast cancer, the BRCA1 gene is something that we look for in individuals who we think might have a predisposition for breast cancer. If individuals carry this gene, then they have a higher likelihood of getting breast cancer than those that don't carry this gene. And so this is an example of a gene that's going to code for a signal that's going to tell the cell cycle to continue very, very quickly. So we can look for oncogenes and see if people have a predisposition to some types of cancer. There are other types of genes. Next to talk about is what's called a proto-oncogene. And that term proto means before. So this is a proto-oncogene, a gene that's not cancerous yet, but it could be. It has the potential to become cancerous. And why is that? Well, a proto-oncogene is going to be a gene that's going to code for positive cell cycle regulation. This, again, is going to be a go signal. But this is something that is going to, in normal healthy cells, be really regulated. So, for example, you have proto-oncogenes that tell cells in your skin to divide when it's time for that cell to divide. An example of this is a gene called the CDK gene, and that is a gene I would like you to know. CDK is a proto-oncogene. But if CDK, for some reason, became mutated, then it could potentially turn into an oncogene. So these proto-oncogenes, because they are normally positive cell cycle regulators, have the potential to become cancerous if, for some reason, there's a mutation. Another type of gene that we want to pay close attention to is what's called a tumor suppressor gene. And these genes do exactly what their name suggests. It looks like humans have built into them information, a code for being able to suppress the formation of tumors. And so what these genes do is they actually will monitor the DNA, and then when they see mutated cells, they're going to intervene and actually prevent those mutated cells from continuing to divide, thereby stopping our cell cycle and preventing cancer from developing. An example of a tumor suppressor gene that I would like you to know is P53. That's the name of the gene. We're actually going to look at this gene more closely to see actually how it works. What I want you to see here, though, is that when we're thinking about cancer, we've got three options: oncogenes, proto-oncogenes, and tumor suppressor genes. In a perfect world, we don't want any oncogenes. We would like to eliminate those. We also would like our proto-oncogenes to work in a way that they are supposed to and never become cancerous. And we also want our tumor suppressor genes to be as healthy as possible, to be able to prevent mutated cells from continuing to divide. Let's look more closely at P53 to see how P53 actually does this. This is a figure that is in your course manual that shows us how P53 actually functions. What P53 does in monitoring the DNA on the inside of a cell is to do a couple of things. If for some reason the DNA becomes damaged in a cell, if the cell cycle displays some abnormalities, or if a cell is experiencing hypoxia, and hypoxia is low oxygen, then those are some events that are going to activate our P53. So right now we're seeing our P53 gene being activated. And when it's activated, it can do a number of things. Let's focus over here on the box on the left. One of the things that the P53 gene can do when it's activated is it can first stop the cell cycle. That's what arrest means. It's going to stop the cell cycle. It's going to attempt to repair the DNA, and if it can do that, it's going to restart the cell cycle, and therefore that cell has essentially been fixed. So when we look at this option over here, the goal of our P53 gene is to fix the cell, and ultimately that's going to maintain stability for that cell. A second option that an activated P53 gene can carry out is something called apoptosis. And this is essentially where the cell will go through some self-destruction. This is for cells that are not repairable. And if cells are not repairable, then we don't want those cells to continue on. So what will happen is the cell will die and the cell will be eliminated, thereby preventing a cell from becoming cancerous. So right here, this is where the prevention comes in. This is how P53 is really preventing a cell from becoming cancerous. So again, both of these pathways lead to the same goal, which is cellular and genetic stability. If I had to summarize in just one phrase, we said that this would be fixing the cell and this would be killing the cell, and either way, our goal here is to keep cancer free, keep the body cancer free. The goal in both of these cases is to prevent a tumor from forming. So we can see clearly how important it would be to make sure that P53 is in good working order. What you can see here is a picture out of your textbook, not in your course manual. And over here on the left-hand side, we can see what we just talked about. The fact that P53 can fix our cell, our P53 could destruct the cell, and ultimately we would have stability. However, if there is a mutation in the P53 gene, then the P53 gene won't be able to work correctly. So that if any of these events here occur in a cell, the DNA gets damaged, or there's some abnormality in the cell cycle, or the cell is exposed to low oxygen, we now know that P53 is not able to intervene and fix it, or to actually destruct the cell. we know that in fact the cell cycle will continue. And this is not what we would like to have occurring, because the cell cycle is continuing now with damaged cells, and ultimately now these cells can become cancerous. So having a mutated p53 gene obviously can lead to cancer, whereas a normal p53 gene is something that we would want. Let's take a picture at how this might look if we looked at this from a cellular perspective. Again, here we can see a normal p53 gene, and in yellow we're able to identify an area where there's been some damage to the DNA. And so in this situation here, the cell was destroyed by that process of apoptosis or cell suicide. However, if we have a p53 protein that's not working and there's damage to the DNA, then we're going to see that the cell cycle is going to continue and continue and continue, and now we have our uncontrolled cell division, and this here is essentially a tumor that's forming, and that, of course, is going to be cancer. Now that we know about the genes that can be involved in the cell cycle and potentially cause cancer, let's talk a little bit about smoking, coming full circle to where we started with our case. I hope you will go to the website that is listed in your course manual and read a little bit about how carcinogens in tobacco actually cause cancer, but I'm going to summarize that topic here. First of all, we want to be clear about what a carcinogen is. Now, carcinogens are things that we can find in a number of different things. They probably are in things we eat, they are in things we may get exposed to, but we know certainly that carcinogens are going to be present inside of cigarette smoke, inside of tobacco smoke. A carcinogen in general is something that causes cancer. That's our definition of a carcinogen. And specifically, we know that carcinogens can actually bind to DNA and cause mutations. So let's make sure we understand this. That means that a carcinogen can move from cigarette smoke or from something we eat or from something we're exposed to and actually cross cell membranes, cross into the nucleus, and attach to DNA and ultimately cause mutations. So I just want to make sure that we're clear that these are things in our environment that are now interacting with our DNA. So one in particular carcinogen to discuss is something called BAP. And this is BAP right here, B-A-P. And I don't care that you know the big chemistry name of it, but it is benzoapyrene. And BAP is an example of just one of many carcinogens that are present in tobacco smoke. There's actually 70 known carcinogens in tobacco smoke, and we're just going to focus on BAP for right now. What's interesting about BAP, it is one of these carcinogens that moves into cells and actually attaches to DNA. And what's interesting about where it attaches is if we follow it closely, if our cigarette smoke goes down into the lungs and we're starting to look at cells, here is the DNA, and as BAP moves into the cell, it's going to bind to the p53 gene. Well, we've already talked about this gene. We know that this p53 gene is supposed to be a good working tumor suppressor gene. But when BAP attaches to this gene and causes a mutation in it, it's essentially knocking out that cell's ability to prevent tumors from forming. So if that cell has some damage to the DNA, and that cell gets exposed to hypoxia, and that cell is going to have abnormalities in the cell cycle, then this is one of those examples where we will have a cell that is going to continue going through the process of cell division. So the scenario that we're looking at is here at the bottom. Here is our non-functional p53 protein, potentially because the carcinogen BAP attaches to DNA, and now it's going to allow for this uncontrolled cell division that we talked about before, and that's ultimately how cancer can occur. So I hope we understand the progression about how having a non-functional p53 gene would ultimately lead to cancer simply because this cell cycle is no longer being monitored by the p53 genes. Another effect that smoking can have on cells is what smoking does to telomeres. So as we look at telomeres in smokers and non-smokers, it's shocking some of the differences that we find. If we were to take two people, let's say, who were 30 years old and actually compare their telomeres, we would find that a non-smoking chromosome would have what we consider normal telomere length for a 30-year-old, let's say. However, if we look at a person who smokes, we can see that these telomeres are much shorter, and that means that aging is occurring. So we know that smoking is also leading to accelerated aging as these telomeres get shortened prematurely. And oftentimes we can see that as certain types of diseases can have an earlier onset in smokers than they might in other people simply because of the age of the chromosomes themselves with regard to the telomere length. I hope that as we've looked at this case, we've understood a lot more how something in our environment could influence our DNA and also be able to influence whether or not a disease is going to be present or not. I'll let you finish up reading about our case with Don and Patricia. I hope what we're going to take from this case as we move forward, though, are some things about DNA structure, DNA replication, our cell cycle, and how things in our environment can interact with our DNA.